This is Professor Mark Dunne from the University of Manchester, who is going to start the program talking about the islet biology in congenital hyperinsulinism.
Thank you very much, Diva. I did wonder what you was going to say then. I thought it might be my accent. Good morning, everybody. Is everybody well? Great. I've got my family with me, so they might appear at any moment. We've been in the States for 2 weeks on a bit of a vacation, so it's a bit sad for me that I've got to leave today because I got lectures first thing on Monday morning, and I couldn't really do it by leaving tomorrow. So that's the only sad thing for me on this occasion. But I'm around this morning. I'm meeting some of the teens, which I'm a little bit daunted about. I'm not sure what that's going to entail just after this. But I will be around until lunchtime. If anybody wants to ask me any questions, then I'm happy to answer them. So I'm from the University of Manchester. I first started research in hyperinsulinism 20 years ago. And I'll actually show you some of the experiments that we did around about that time, because I've been asked to talk about islet biology, which I will do, but I also want to sort of illustrate one or two aspects of research, things we did a long time ago, things we're still doing, and things that we've done very recently in the lab as well. I don't want to make it too heavy. I'm not speaking to the experts in the room, so if anybody has any questions at any time, please put your hand up. The University of Manchester is very close to the Children's Hospital in Manchester. There are 3 of our clinical team here. They're all sitting there looking at the screen at the moment. Stand up. There's Indy, there's Maria, and there's Lindsay there. So please talk to those during the meeting, and all 3 of them are going to be participating as well. Islet biology. It's hard to know where to start in some respects. But you'll all be aware that the pancreas is the problem in children, your children, and children with congenital hyperinsulinism. And the functional unit, the endocrine part of the pancreas, resides in these structures here, the islets of Langerhans. And they are found throughout the pancreas. There is somewhere around 1 million of these islets that sit within the pancreas. They're connected intimately with the blood supply to the pancreas. They've got nerves which communicate to and from the pancreas. But they're distinct, they're different to the other things that the pancreas does, which is to help with the digestion of foodstuffs. Despite the fact that they are an incredibly important part of the control of blood sugar levels, only about 1% of the tissue, the whole pancreas, has the islets. So the islets are the tiniest proportion of the cells that reside within the pancreas. And that has some challenges when we think about research. They're about 0.1 millimeter in size. You can just about see them with the naked eye if you isolate them. And here's an image of some isolated islets. And it's tricky to get preparations of islets like this out of the pancreas because they're embedded within the pancreas, and there are so many of them, and they're distributed throughout the pancreas. You need certain techniques and skills in order to be able to tease them away and to basically destroy the rest of the pancreas, but leave these structures intact. As I say, there's about 1 million in the human pancreas. Each circular structure contains around about 1,000 to 1,500 cells, and there are a number of different cell types within the individual islets. There are the insulin-secreting beta cells, there are the glucagon-secreting alpha cells, and there's also the delta cells which release somatostatin. Some of you will be aware of octreotide, which is a mimic of endogenous somatostatin and is used in the treatment of hyperinsulinism. But there's also two other cell types as well, ghrelin and pancreatic polypeptide. So all these cells sit within these capsules, these mini organ structures, and because they're sitting next to each other, they begin to talk and communicate with each other. And when we think about the control of blood sugar levels, we need to think not just about an individual cell, but how that cell communicates with its other cells, and how that islet is then positioned within the pancreas next to the blood supply to the tissue. But I'm really going to concentrate mainly upon the insulin-secreting cell, and I'll also mention the glucagon-secreting cell in the context of control of blood sugar. And it's the endocrine units, the islets of Langerhans, that really just have a major core role in controlling blood sugar levels. And their jobs is really to detect blood sugar and to respond to that blood sugar level. And the two principal cell types that I just want to introduce in a little bit more detail are the beta cells and the alpha cells. So the beta cells release insulin. They are built to release insulin. They release somewhere in the region of 15,000 granules per day, and they should keep on going for as long as an individual lives. People with type 1 diabetes or type 2 diabetes have problems with the production of insulin. Children with congenital hyperinsulinism produce too much insulin. But what insulin does as a hormone is it allows us to use glucose, and it allows us to put glucose into tissue so that it can be stored as glycogen. And that's the two main principal effects of insulin in the control of blood sugar levels. And glucagon acts in the other way. It needs to detect low sugar, and it needs to release, the alpha cells work the other way. They need to detect low sugar. They release glucagon, and glucagon's then used to move storage sites of glucose back to the circulation. So glucagon allows us to convert glycogen back to glucose so that it can re-enter the circulation. So we have two cells which are found right next to each other in the islets of Langerhans that both detect and respond to glucose but have completely opposite physiological actions. And we focus mainly upon the beta cell in congenital hyperinsulinism, but the gene defects that are carried by most children with congenital hyperinsulinism also affect the alpha cell as well. And that's an area of activity that a number of researchers are moving towards. So that's a little bit about what they do when they've detected glucose. I want to talk a little bit more about those detection mechanisms. And the reason why I want to focus on those a little bit is that this is one of the areas in which many of the mutations that affect your children are causing problems with the system. So let's just focus a little bit on the beta cell first. The beta cell is able to detect rises in blood sugar levels, and glucose acts as a principal stimulator for the beta cell. And it's that connection between glucose and insulin that many of us are investigating and are interested in. And a key part of that detection and response system involves changes in the electrical activity of the cells. So insulin-secreting cells are they're like nerve cells, they're like muscle cells, in that they have electrical properties. I've kind of tried to illustrate that by a series of lights and a sort of a blue shock there. And what we know in terms of signaling in cells is that glucose needs to come up inside the beta cell. There then needs to be a change in the electrical activity of that cell, and that electrical activity is then responsible for driving more signals so that insulin can come out of the insulin-secreting cell. So the coupling mechanisms involve metabolism and electrical activity. And you have the same kind of features taking place in the alpha cell. But what glucose does to the alpha cell is, once glucose levels are raised, the alpha cell begins to be switched off. And the beta cell further switches off or inhibits the alpha cell. So we have glucose having a positive effect, if you like, on the beta cell and a negative effect on the alpha cell. And the beta cell also is throwing things towards the alpha cell and switching it off further. Because the last thing you want to do if blood sugar levels are high is to get glucagon out, so that because glucagon wants to raise blood sugar levels. So it's a kind of an interesting balancing position that takes place within the islets. What happens when blood glucose levels begin to drop is that electrical signal switches off. And so in the presence of low sugar levels, the beta cell will cease to be electrically active, and the signals which drive insulin out then stop. And it's that control of electrical activity, almost like a switch, which is a problem in children with congenital hyperinsulinism, because the lights stay on in children with CHI, whereas in healthy children, those beta cells cease electrical activity once glucose levels are lowered. When glucose levels fall, the alpha cells become activated. So you've got a decrease in a physiological signal that is stimulating these cells. And as a consequence of that decrease in the physiological signal, the alpha cell will then release glucagon, and it will cause the liver glycogen stores to be mobilized. And here's the position that we have with children with congenital hyperinsulinism. With CHI, the beta cells are constantly electrically active. They can't switch off their electrical properties. And as a consequence of that, low blood sugar levels are not connected to the beta cell, and insulin further drives down those blood sugar levels. So we get into a cycle of decreasing low sugar levels because insulin is still coming out of the beta cell and it's having its normal physiological effects upon target tissues. Yet insulin is not switching off, glucose is not regulating the beta cell. And the further problem that your children have is that the low sugar levels are blind, the alpha cells are blind to the low sugar levels. And that impact of the beta cell on the alpha cell is still taking place. So the alpha cell really can't respond in children with congenital hyperinsulinism because they carry some of the genetic defects, and the beta cell is pouring out insulin and other influences and keeping the alpha cell suppressed. And this is the reason why children with CHI have problems in mounting normal counter-regulatory control mechanisms. And we've been interested for a number of years in how the genetic problems impact upon the insulin-secreting cell. Because in the absence of any way to switch off the beta cell, the problem, as you all know, is that low blood sugar levels are driven further and further down, and this has major implications for your children. You'll hear from Sean Ellard about the genetics of congenital hyperinsulinism. You'll hear from Charles, and I think Sean as well, about the fact that some of these genetic lesions affect just a small proportion of the pancreatic tissue. So you have focal disease, where there's a small lesion of hyperactive insulin-secreting cells, but the remainder of the pancreas is unaffected. And we'll also hear more about the diffuse form of hyperinsulinism, where all of the islets in the pancreas are affected and the lights really stay on in all of those tissues. So in terms of research in the context then of islet physiology, I just wanted to give you a couple of examples of the kind of things that we've done and we do, and what other people do, both kind of from a few years ago and contemporary experiments. and things that we're currently doing right now in the lab. If you want to try to understand what is going wrong with the system, the easiest way is to get access to that tissue and study the physiology of it in the laboratory. And at one level, that ease at which we can study the diseased tissue is made easy in congenital hyperinsulinism, because many children have pancreatectomies in order to prevent hypoglycemia. And we can gain access to tissue post-surgery, and we can, in principle, isolate islets from that particular tissue. But there's a few challenges along the way. One of them is that we need a regulatory compliance in terms of your permission to do research, and institutional and regional permission to perform studies. We also need to have in place the proximity. One of the problems with working on the pancreas is the pancreas is also involved in digesting foodstuffs. And if you take the pancreas out of the body in terms of the operation, the pancreas itself will then begin to digest its own tissue. And in order to be able to produce healthy insulin-secreting cells, you need to have a kind of a rapid way of moving tissue from the surgical environment to the research environment. However, you've always got to come through the pathology department, and the histopathologists are generally, in many places, happy to involve and engage in research. But they still have a job to do. They still have to be able to diagnose what the problem with the tissue is. So we work very closely with our histopathologist, and I know it's the same here at CHOP, in order to make the movement of the tissue out of the operating theatre into the research laboratory as smooth as possible. And then you've got some problems with the isolation of the tissue. Usually the surgeons are incredibly skilled, and they just take out the right amount of tissue that's necessary, and that can impact upon what you're doing in the laboratory. And also, at the end of the day, we've got to destroy most of the tissue if you want to get the islets of Langerhans out intact, and that can be problematic. But for many of us, the effort is really worth pushing because of the impact that basic research can have on this particular condition. And another problem in this area is that if you're only dealing with a small amount of tissue from a small pancreas from small children, there's only a certain number of things that you can actually do with that tissue at the end of the day. You can't do major biochemical types of assays because there's simply not enough tissue available for research. Now, we started our journey in congenital hyperinsulinism 20 years ago by doing some experiments on tissue that came from children following surgery with Great Ormond Street Hospital. And in those experiments, what we were doing, and what we still do, is we are able to isolate single insulin-producing cells from the tissue, and we can place against the cell membrane a small suction pipette, which has the tip diameter of less than the size of a human hair, and is a very smooth end. So it's not a needle that we push into the cell, it's a small suction pipette that sits on top of the cell membrane. And you can see here, if you apply that suction pipette to the isolated cell, you can see that small little piece of tissue, small little piece of membrane that's sucked up inside the patch clamp pipette. And this technique is really kind of useful because it means that we can work on single cells, so we get over the problem of access to limited tissue. But more importantly, it gives us a real window on what is happening in the insulin-secreting cell. So the electrophysiology really did unlock the basic problem that the beta cell faces in the congenital hyperinsulinism tissue. It allows us to link the clinical profiles with the genetic profiles. And I just illustrated here one or two experiments. The first one in the upper panel will be from a a healthy human beta cell. The one in the lower panel here in the red box is what we would see with from a child with congenital hyperinsulinism. And the kind of experiments that we do are fun experiments because the tissue's alive and you get data in real time. And we were looking here at what diazoxide does, and we were looking here at what the physiological regulators of these particular ion channels are doing. And here's what the data looks like if you have a control healthy beta cell. And it's these little squiggles here that are associated with electrical changes, and these are part of the switching mechanisms that the beta cell uses to turn on and turn off electrical activity. And you can see here this healthy beta cell has a really good response to diazoxide, and that response there would switch off that electrical activity in the beta cell and stop insulin secretion. And that's how diazoxide works. It turns on this particular ion channel, and this particular ion channel is controlling the electrical switches in the beta cell. And the recordings from the child here who has congenital hyperinsulinism couldn't be any different. What we're looking at is essentially a flat line. And when we're doing these experiments in the lab, and you're looking at flat lines over and over again, it kind of, it's kind of fun in some respects because we're actually describing the basic physiological problem of congenital hyperinsulinism. And so what we now know is that the genetic lesions, the problems that are associated with congenital hyperinsulinism prevent these channels being made, and because they're not being made, the beta cells cannot control their electrical activity. And we've done those kind of recordings on many patient tissues. And it allowed us to suggest and propose this model to the scientific and the clinical community, which has been adopted now and is the cornerstone of understanding congenital hyperinsulinism. This particular ion channel is called the ATP-sensitive potassium channel. It's encoded by the genes ABCC8 and KCNJ11, which Sean Ellard will talk about later. In children with CHI, it doesn't work. It doesn't get to the cell membrane, or it doesn't biochemically function properly. And these cells are electrically active in the absence of any physiological stimuli. And the coupling then between electrical responses and calcium coming in and insulin going out are pretty easy to understand, because calcium is a powerful signal to the insulin secreting cell to push insulin out. And this happens in the absence of any glucose signaling. So these types of experiments allow us to understand how the genetics links to the biology of the beta cell. They explain how ion channel defects can lead to hypoglycemic events. They also allow us to understand how many patients have poor responses to diazoxide. Many of your children will be treated with diazoxide, and diazoxide acts on a defective protein. It kind of doesn't make sense. From a drug development perspective, I guess John Kinzel over there will tell you, why would you design a drug that is not, and the target is the core problem in the disease? If you started from scratch, you wouldn't use diazoxide. But it does have some effectiveness because not all the patients have a complete problem with this particular channel. This type of model also allows us to think about opportunities for identifying new drug targets within the pancreatic beta cell as well. So what are we doing in some of our research activities at the moment? We're still fundamentally interested in the beta cell, but our surgeons in Manchester and across the planet are getting better and better, and the amount of tissue that they sometimes have available for research becomes quite limiting, particularly in the focal form of the disease. So here's a few experiments that I was just going to show you involving a new technique, a technique called serial block scanning electron microscopy, which is kind of quite a mouthful. And here's the amount of tissue that we got from the surgeon in this particular case of congenital hyperinsulinism. It came from a child with focal disease. It's kind of been embedded in resin. That's why it looks dark coloured here. But you can see from the PhD student's fingertips the size of the tissue that we're dealing with. And we've put it in resin, and we've then shaped this resin block so it's got a flat face on it. And what we then do is we place it in a machine which cuts thin sections of tissue through the resin. The resin keeps the tissue intact. And then we're able to produce electron microscopy studies on those very thin sections of tissue. The tissue sections are 20 nanometers thick, so incredibly fine pieces of tissue. And we can take somewhere in the region of 500 sections or more from a piece of tissue. And that means we can stack the images up. And if you can stack the images up like this, you can also then produce movies which allow you to look at the tissue, not just on the surface, but to walk through the tissue. And I know Devlin likes these movies, so I'm going to put it on again. This is the second time I think I've shown this movie to Devlin. So you're looking here at a walkthrough of a tissue. It sort of settles down in a moment. It's probably easiest to see the blood moving through the capillaries. Let me just get this up here. Here's a blood vessel, and these squishy looking things here, they're the red blood cells, and they're all compressed in together. The cells here that are sitting right next to that blood vessel, these are the insulin secreting cells, and they are constantly releasing insulin. That's why the secretory packages look pretty empty of insulin. But the red blood cells are kind of quite fun to look at, and it really shows you how slow the blood is moving through the islet, and how the red blood cells are all changing their shapes because they're compressed in a very small space. At any one moment in time, 20% of your blood is in the islets. So there's a huge, rich blood supply going to the islets. And the beta cells and the alpha cells are all connecting with that blood vessel. The things here, these sort of grey structures, they're the nuclei. So there's a single nucleus. That's a nuclei as we're chopping through it, and the cell structure's somewhere around here. And eventually these two pipes here join together, and it's kind of almost where there's a sort of U shape through the tissue. And you can see here now the capillary has got together, and the red blood cells continue to sort of begin to move through that tissue. So why are we doing that? Why are we doing these kind of experiments? Well, one thing you can do with this type of technique is you can sort of physically look at how the tissue's put together, but you can also begin to digitize it. You can begin to convert these photographic plates into three-dimensional structures by digitizing the images. And you do this simply either by hand or by getting the computer to draw around a shape of particular interest. And you do it on one photograph, then you go through and you do it on the next photograph. And you can begin to build up a three-dimensional structure of the tissue. So here we've reconstructed some of the blood supply associated with an islet, the capillary networks, and we've illustrated here inside it the red blood cells, and you can see how closely packed in they are. And this blood vessel here just loops its way through the islet, and we can time. Yes, I'm nearly finished. Sorry? Yeah, I was just going to show one more thing. The other thing that we're doing is we are looking at the connectivity between the insulin secreting cells and the blood vessels. And a little bit of research here, we've been able to quantify, to count the number of mitochondria that are inside the insulin secreting cells and the alpha cells. And the reason why that's important is because there are big differences between the energy sources in the hyperinsulinism beta cell and control beta cells. So I'm finished. Thank you very much, Diva. I'm happy to take any questions. and I'll leave that model up there. Thank you.
Thank you very much. I told you he had beautiful pictures. So we have time probably for a couple of questions. If anyone has any question for Professor Elder, Professor Mark.
Here, I have a mic.
Thank you. Hi, I'm curious. When you talk about the ion channels that aren't functioning, is it a materials problem in the way the islets are constructed because of the genetic defect, or an electrical problem in that the biology is fine, but the electrical circuit isn't working well enough?
It's a really good question. There is not the genes that encode for the ion channels. They have a large number of different mutations. They're big genes. They're bigger than the cystic fibrosis gene, and there's a large number of different mutations that give rise to the same clinical problem. And some of those mutations stop the channel being made. Some of them stop it getting to the cell membrane. Some of them stop the movement of ions through the channel, and some of them stop that channel from detecting the biochemical changes that glucose causes the beta cell. So there's not one particular mutation. The common output is the electrical control is disturbed. So it's a really good question, and the genotype-phenotype correlations are something that a lot of investigators are looking at.
Sorry, and do you have a sort of distribution of what that looks like in terms of each of those points of failure that you just mentioned?
Yeah, I do. I haven't got it here, but we can count the number of channels that sit on the cell membrane, and in about 20% of the patients, there's no channels whatsoever, like the one I showed. In 80%, there is something, but not enough to help the cell to control. And I think, looking forward in the future, I don't know whether you're following the cystic fibrosis field of research, but new drugs have been developed that help CFTR get to the cell membrane. And those types of drugs could be potentially useful in hyperinsulinism. We published a paper about 4 years ago showing in a series of 8 patients that you could recover channels by similar procedures in vitro. So we're kind of hopeful that that type of approach will be effective for a certain number of patients. And if we know what the genetics is beforehand and we know what that genetic problem is, we might be able to streamline certain groups of patients and suggest that let's try that drug as a trial because it may well work.
We have another question here, and I think we're going to stop there.
Sure. You pointed out that it's odd for diazoxide to be the target for the potassium channel. Obviously, the calcium channel is working fine for this. Is there a reason why we don't go after the calcium channel, or is that something else that somebody's working on now?
No, I think we published a paper 20 years ago when we did this basic physiology, saying, look, the calcium channel here could be worth it. And a colleague at Great Ormond Street took a chance and tried nifedipine on a patient, and it gave that child some benefit. And we talked about this at the scientific meeting, didn't we? There are odd groups, not odd groups. There are groups around in different communities, perhaps not in the States, but elsewhere internationally, that have tried nifedipine once or twice and reported positive and negative case reports. The problem with nifedipine and the beta cell voltage-operated calcium channel is that the same channel is present in the vasculature, it's present in the heart, it's present in the neurons. So unlike sort of treatments for aneurysms or cerebral aneurysms, you can't selectively hit the beta cell voltage-operated calcium channel in the same way. But there's a sort of feeling that it might give some benefit in combination with other drugs. So it has been thought about, but it's mainly the off-target effects that have prevented wide use of it.
Okay, thank you very much.
Thank you.